So, welcome back to the virtual classroom. Today I am going to be speaking about the cell cycle and the regulation of the steps within it. Okay. So how is it started? How is it timed? How does it physically happen? And what is the function of it? Right? Why do we have the cell cycle? Specifically today, I'm going to be looking at mitosis. We will leave meiosis for the genetics/inheritance storyline. Right? And so we will be looking at just the mitotic processes today. Like meiosis and mitosis are very similar in nature. So a lot of the storyline that applies to mitosis will also be valid for meiosis. Okay? So these are the things I'm going to talk about today. Overview of the cell cycle, how CDK and cyclin regulate the steps of the cell cycle through what are called checkpoints, okay? How the kinases actually work, and what happens if this process goes awry. Right? What are the bad endings? So that's what today's video is about. So, on the board behind me, I have a representation of the general cell cycle. This should look familiar to everybody. You would have encountered this in Biology 1. We've touched upon it in class so far this year, so this should not be a mystery for anyone. Okay? But just a quick recap, just in case. Okay? The M phase is the mitosis or meiosis phase. Right? Ultimately, it's the end step of this whole cycle, but because it's cyclical, right, the end is also the beginning. Right? And so in this storyline of mitosis, this is mitosis. Okay? When it finishes, I now have two new cells ready to do their cell life stuff. Okay? And so immediately upon exiting mitosis, the cell should enter what is called G0. Okay? Growth phase 0. It's a state of stasis for the cell. Okay? It's not going to be dividing anytime soon, so it doesn't need to grow at all. The cell just needs to do its job. So whatever this cell's job is, it's chilling in G0 and doing that job. So as adults, most of our cells, most of the time, are in G0. Upon an event that triggers the cell to enter the cell cycle to begin cell division, right? We will move from G0 into G1. Okay? And now the checkpoint steps are something I'm going to talk about in the next section of this video. Right? So we'll cover what that actually means. But for now, just know if the checkpoint is hit, we move from G0 into G1, growth phase 1. And now growth phase 1 is where the cell is gaining in size. Right? We're adding more cytoplasm, we're adding more organelles, we're adding more of our functional proteins. Because right? Ultimately this cell, at the end of the story, is going to get split into two. So it effectively needs twice as much stuff in order to be split in half without being ruined. Right? That makes sense. So growth phase 1 is where we're adding most of those materials. Okay, cell's getting nice and big. She's a chunky girl now. Okay? After that completes and the checkpoint of G1 is hit, the cell moves into S phase, which is DNA synthesis. Right? And so this is where the DNA is being replicated. So every single one of the chromosomes that this cell has is being copied over in an identical form. So the cell goes through DNA replication, and it now has two copies of every piece of its DNA. This is important again because it's about to split in two, and each of those daughter cells needs a set of DNA. So by the end of DNA synthesis, I have two identical copies of my DNA. And so if this is my representation of a chromosome prior to S phase, post S phase, this is what I'm looking at. And now this model is not accurate because these are mitotic chromosomes. right? The bundled shape doesn't happen until we hit the phases of mitosis. And so during S phase, the DNA is actually long and striggly and DNA, right? So that double helix, just a long strand of that double helix DNA shape. Okay? And so I have those 2 copies, and they're stuck together in a couple different places. They just float around. Okay? Once I have my 2 copies of my DNA, we move into growth phase 2. Right. And growth phase 2 is adding a couple more organelles, making sure everything's all set to be split up, make sure I got enough of everything. Okay. It also is where the centrosome is put together. Right? And so the centrosome is an organelle that is very important in the mechanics of mitosis or meiosis. Right? It's where the microtubules are originating from. And so you get these 2 like anchor points on either side of the cell, and they have these big long strands that come out and find the DNA chromosomes and grab onto them and pull them around and move them where they need to be. Okay. So the centrosome is getting made in G2, getting ready to do this thing that it does. Once all of everything is made and the cell is ready to be split in half, okay, it moves into M phase, which again is mitosis. And the very next section of this video will just be an outline of the steps of mitosis. Right? But the goal of mitosis is specifically taking these 2 sets of DNA, boom, that are identical copies, ripping them in half, and moving them in an organized fashion to either side of the cell, so that when we cut that cell in half, both halves of the cell have a full set of DNA. That's what mitosis is. It's the splitting of those chromosomes into even halves. Okay? Cell cycle. M, G0, G1, S, G2, M. Cyclical. Around and around and around. So, on the board is our good old friend P mat, right? Like a mat upon which one would urinate. A P mat. Catches the P. Easy acronym to remember the steps of mitosis. Okay. And so we have, the first step is prophase. And in prophase, we are taking the nucleus that is surrounding the DNA, and we're breaking it apart. Right? And so we need to be able to move the DNA throughout this whole entirety of the cell, and the nucleus is obviously in the way when we try to do that. So we just break the nuclear envelope open, get rid of it, it dissolves, bye. Okay. And then we condense the DNA into mitotic chromosome shapes. Right? And so as I said a little bit earlier, typically the DNA are these long strands. Okay? That's really hard to move around in an organized fashion. So we coil up the DNA around proteins called histones, and we get them into these tight little balls, okay, of organized DNA sets that we typically think of when we think of the shape of chromosomes. Okay? So these coiled DNA strands are mitotic chromosomes. Okay? When it's out and stringy, it's still a chromosome. Okay? It's just not as organized. And so we condense them into these mitotic chromosomes. They're ready to be moved. Okay? Once that is complete, we move into metaphase. Okay? And metaphase takes the organized condensed chromosomes, right? The centrosome sends out its microtubules, and they just float around until they can connect with one of these condensed chromosomes. Okay? And so it connects in a spot called the centromere, okay? And specifically to regions called the kinetochore. So each of these chromosomes have this kinetochore region, okay? And the microtubules have reached out, okay? And they've grabbed this kinetochore. Okay? And so the kinetochore has little shapes that stick out. They match the microtubule shapes. When they touch, they lock together. And now they're locked. Right? And so each of these microtubules is stranding out from the centrosome, looking for a chromosome to grab onto. Okay? And just through random chance, eventually they bump into each other and they lock. Now, once that happens, right, if I lock microtubules onto this side of the kinetochore, this side is going to be looking for some microtubules as well, okay? But the shape is slightly changed. Okay? And so that way it will only lock in with microtubules from the opposite centrosome. Okay? So there's a centrosome on each end of the cell, sending out their little grabbies. Okay, and it will lock into one when it finds it, okay? And then I want a centrosome from the other side to lock into my opposite kinetochore. So it changes the shape a little bit so that this kinetochore can only attach to the opposite poles microtubules. Okay? Prior to that, it can connect to either one. Okay? Boop, they lock in, cool. Once the centrosomes have all locked in to the chromosomes, right? Everybody's happy, everybody's cool. The kinetochores can sense that everybody's locked in, right? And so there's a certain amount of pull depending upon how many chromosomes are locked up, right? And when it hits just the right amount of pull, the kinetochores release some proteins, okay? And that triggers the centrosomes to move every chromosome right into the middle of the cell, right? And so there's this equal pull on both sides, and that gets everybody right in the middle. So all the chromosomes are lined up down the middle of the cell. That is metaphase. Okay? And again, once everybody's lined up down the middle of the cell, okay, that creates a very specific amount of tension on each half of the kinetochore. The kinetochore changes shape due to that very specific amount of tension. Okay? And it splits the centrosome that's holding it together. So the centrosome is broken open, boop, okay? And now these microtubules are still pulling, right? So once that centrosome is split open, these two chromatids separate, and they get pulled towards the centromeres. And so now I have a copy of each DNA set, each chromatid, which are now individual chromosomes, on either half of the cell. That was anaphase. Okay? Anaphase is the splitting apart. Once I have my even halves on both sides, telophase is triggered, and that recreates the nuclear envelope around our sets of DNA. So both of my DNA sets get an envelope around them. They are now two little nucleuses. Right? So for a little bit of time, this one cell has two nucleuses. And once that's finished, cytokinesis is triggered. And cytokinesis is the process of actually taking the entire cell, cutting it in half, and plopping it into two new daughter cells. And the two new daughter cells are then going to enter G0, move into G1 eventually, and get ready to go. Okay? So that's mitosis. Prophase, metaphase, anaphase, telophase. The function is to take one parent cell and to generate two identical daughter cells. We need to do this if we need to grow or if we need to repair damaged tissues. So, back to the general cell cycle for a moment for a more detailed look at how the steps are regulated. And now the regulation is controlled via cellular communication mechanisms for the most part. Right? It's triggered initially through cellular communication. And so shifting the cell from G0 to G1, there is what's called a checkpoint. Certain conditions must be met in order for the mechanisms to be triggered. And in this case, the primary condition that must be met is that the cell must have this molecule cyclin B. Okay? So cyclin B must be present in order to move from G0 to G1. And the way that cyclin B is generated for this cell, okay, is through a cellular communication mechanism, right? And so we have to keep in mind what purpose this cell cycle has. Okay? And now, again, as adults, most of the time we want to divide our cells when we have been injured, right? Some tissue has been destroyed, okay? Whether it just be through time and degradation or through an actual trauma, right? We have damaged cells, we want to replace them with happy cells. And so here I have two cells of the same tissue. Okay? So maybe they're muscle cells, right? This is in the bicep. Let's call these bicep cells. Okay? Something has happened to damage one of these cells. Okay? This cell is now ruined. Okay? That process of ruining the cell triggers mechanisms within the cell to generate and release ligands. And those ligands are specifically going to be called growth factors. Okay. So these growth factors get released into the interstitial fluid around this damaged cell, okay, and they reach the tissues nearby. So when they reach other cells that are still healthy and haven't been damaged by this trauma, those cells will receive those ligands. A transduction pathway is triggered, and ultimately there is a response by this cell. The response by this cell in this particular case is to generate the cyclin B molecule, right? And so I make this cyclin B when I've received these growth factors from the cells around me. Once cyclin B exists, okay, it will float around, okay, and eventually it will find this other molecule, this enzyme, that is constantly in the cell. Okay? It is a CDK, cyclin-dependent kinase. Okay? And so kinases are types of enzymes that do very specific jobs within the cell. Okay? And the cyclin will fit into the enzyme. It will turn the enzyme on, and that enzyme will now do its job. This enzyme's job is specifically to phosphorylate other proteins within the cell. And so if this particular checkpoint is to move from G0, where I'm not doing anything for cell division, into G1, growth phase 1, where I'm growing, I'm adding more cytoplasm, I'm adding more organelles, right? This particular interaction of cyclin B and CDK will phosphorylate the proteins that mechanically add more cytoplasm and trigger the building of new organelles. Okay. So that is how kinases are actually working. They are phosphorylating other proteins. They're effectively turning on the other proteins who are just floating around and waiting to do their job, right? They're almost the right shape, but they're not quite the right shape yet. Okay. You slap a phosphate on them, they change shape a little bit, and they're doing their job. In this case, their job is to say, hey, cell, cytoplasm time. Let's make more cytoplasm. More cytoplasm. Hey, mitochondria, we need more of you. Let's make more of you. More mitochondria. Right? And so that's what these CDKs are doing. Phosphorylating other proteins who will specifically do the jobs that are needed at hand. Okay? As soon as the cyclin engages with the CDK, the cyclin changes shape slightly. It becomes a little bit more unstable, and it will slowly begin to degrade. It will fall apart. Okay? Eventually, it falls apart to a degree where it doesn't fit in the CDK anymore. It just falls out. Boop! Okay? So without its cyclin, the CDK stops phosphorylating anything, right? And whatever mechanisms were triggered by this initial engagement are now stopped. During that whole process, another cyclin would have been built, okay? And so now the CDK is empty, okay? And the new cyclin can fill in the gap. So the CDK can start phosphorylating things again. This time it's going to phosphorylate different proteins. Okay? And so if I'm moving from G1 to S, where I want to do DNA replication, it's going to phosphorylate the proteins that run the processes of DNA replication. Right, so I'm going to turn on those proteins, and DNA replication is going to begin. And we're going to do the same process over again. Right? That cyclin is going to start falling apart Okay? Eventually it falls out, okay? And a new cyclin fills it in, and we can do the mechanisms of G2. That cyclin falls out, and a new cyclin falls in, and we can start mitosis. Okay? So that's how CDK, cyclin-dependent kinase, regulates the mechanisms and the time frame of the cell cycle, right? So it adds a timer to it so that these stages are happening for the right amount of time. Because if they happen too long or too short, the cell isn't going to be ready to do division. We either have not enough stuff or too much stuff. And either way is not good. Right? So these CDKs are very important. Right? There is a lot of other protein cascade creation channels involved in this story. I'll put a picture of some up here, right, just to give you an indication of how many of these conditions are actually needed to regulate the cell cycle. But for the sake of our understanding of the principle and the mechanism, right, we can just focus on the CDK aspect of it. Okay? CDK. I love CDK. So I already filmed this one, and then I accidentally deleted it. So I got to film it again. What happens when a cell regulation mechanism goes awry? And so typically, our regulation mechanism is very, very effective, right? We have each of these checkpoint steps throughout the cell cycle and within mitosis itself that requires a very specific concentration of many different molecules. Right? And so if we look at a shift from G0 to G1, right, we need that cyclin B in order to make that happen. But in order to make that cyclin B, I need the growth factors from outside the cell. Right? And I also need all of the precursors that are required to make cyclin B. Right? And so to make cyclin B might be a 18-step transduction pathway. Right? And if not, all of those steps are met very specifically, I will never get cyclin B, and I will never kick off the cell cycle. And so we can create very specific conditions required for any of these steps to occur. And that's true at any of the checkpoints. And now the checkpoint from G0 to G1, it's cool if it's not met, because then the cell just hangs out, right, and keeps doing what it's done. The checkpoints from G1 to S, from S to G2, and G2 to M, if those go wrong, it's not so easy just to stop and pause and wait. Right? Because either the cell will have too much stuff to be good, or the cell won't have enough stuff to be good, and it's just not going to go great at all. Right? Something's going to go wrong if these steps are not done correctly. So there are mechanisms to check if each of these steps are done correctly. And if something goes awry, like something's not right, the cell will trigger apoptosis. A bunch of lysosomes will release all of their digestive goos, break down the cell will explode, right? And all those goodies will be released into the interstitial fluid as resources for the cells around it. Right? So if a cell is not doing its job, if it's doing a poor job at its life, it's not doing cell division properly, right? It will just go away, and the other cells that are doing their job properly will get those resources. And so that's the ideal situation. Right? The alternative is that our regulation mechanisms break. And that happens, right? And so typically how that happens is there's a genetic mutation in one of our cells that changes the shape of one of our regulatory molecules, right? So let's pretend it's the CDK between G0 and G1. And so the mutation might modify the shape of CDK so that when cyclin B engages with it, it never degrades, it doesn't fall out, right? It just stays as one unit. So I never get that timer to stop my process, right? And so this cell might just keep going through cell cycle over and over and over again and never stop in G0. Okay? And this out of control, excessive reproduction of cells is what cancer is, right? That's cancer. One of our cells has gone buck wild and just keeps copying itself over and over and over again. Right? It never stops to do its job. It just keeps copying and copying and copying. And now typically our body has an immune response that will say, hey, you're not doing a good job, I'm going to kill you, right, and kill it. But sometimes the cancer gets ahead of us, right? And one cell very quickly becomes 2 cells, and 2 cells very quickly becomes 4 cells, and 4 cells very quickly becomes 8 cells, so forth and so on. And so it's easy to see how that can get out of control, right? Our body can't keep up. And it's really hard to treat cancers because the cells are technically our own cells, right? They're not a foreign invader or anything like that. And so a lot of the things that will hurt those cancer cells will also hurt regular cells, right? And so we are currently looking at like gene therapies and specific receptors for specific types of cancer. Right? And so some of these modifications that cause the cancer can also be used to target the treatment of the cancer. But yeah, that's what happens when our regulation mechanisms go crazy. Everything breaks and it's not good. Cool. And that's it. Done. Finito. Finea. Finissant. We finished. Okay. Cell cycle. M, G0, G1, S, G2, M, over and over and over again. Mitosis creates 2 identical daughter cells by dividing duplicated DNA into equal halves via some very cool physical mechanisms of pulley thingies using centrosomes and microtubules. Right? These steps are regulated through CDK and other kinases that allow for a very specific timeline, both in arrangement of mechanisms and in duration of mechanisms, right, that allow for a very specific outcome and very regulated, highly effective mechanism to ensure that each of our new daughter cells are exactly what they are supposed to be. That's it. Bye.